Hello, welcome to R.K Medicos. In this section, we are going to discuss about the biochemistry part 5, that is general biochemistry basically. And we are going to see here the enzymes and the classification of enzymes and what are the different types of enzyme we are going to see here and how they are going to perform their action, what are the different types of chemicals or the enzymes which are included in the different types or the classes of the enzymes. So let's start and who's the classify these all enzymes in the different classes. The first one is International Union of Biochemistry and Molecular Biology basically, that is also called as the IUBMB, and they are basically classify initially the 6 classes of enzymes, but now one recently added in 2018 August, one new classes is added in the enzymes, that one is translocase basically, and one new classes will be added. We can see here translocase, that is on the 7th number. The enzyme classes will be basically 7 now, and many books there is also written 6 classes, but for now there are 7. And we can see the first one is the trick for it, that is for the memorizing the classes or the names of these enzymes. OTHLIL, we can write like this, OTH lil and T. You have to memorize it, OTHLIL, and after that you can memorize T from anything. That is, these are the 7 classes of the enzymes basically. The first one is oxidoreductase. O stands for oxidoreductase, T stands for transferase, H stands for hydrolase, L stands for lyase, isomerase, ligase, and T stands for translocase. Basically these are the 7 classes of enzymes we are going to discuss here, and we are going to discuss one by one of them. So first we are going to see which one, oxide reductase. And the class first, oxidoreductase, what happened to these enzymes, what they are going to do, they are catalyzes the oxidation and reduction of any reaction. They are going to catalyze the oxidation and reduction of any reaction. And what are the different types of oxidoreductase reactions, you know? There are different types of classes they divided. The first class, oxidoreductase, is divided into subclasses. And what are the subclasses? There are basically 6 subclasses here. We can see dehydrogenase enzyme of groups are included in the first class. All the oxidase enzyme is included in the first class. Oxygenase in the first class. Peroxidase enzymes group will be in the first class. Catalase will be also included into the first class, and reductase. So these all 6 different types of enzymes are the classes of which first class, and for name of the first class is oxidoreductase. They are basically going to do oxidation and reduction reactions here. That these all are the enzymes what we see here. The subclasses will be dehydrogenase, oxidase, oxygenase, or the peroxidase, catalase, and reductase. These all are included inside the one single group. That one is called as oxidoreductase. They are going to do oxidation or the reduction. We can see several examples one by one. For the first, we are going to see in the peroxidase, there is one example is important, glutathione peroxidase. We discuss later what are the role of glutathione in our biochemistry or in general life, but for now you have to memorize the peroxidase enzyme is basically glutathione peroxidase. In our general life it's important. Catalase enzyme, where it's located in our cell, that is in peroxisomes. In the peroxisome part of the cell organelle they are located, that is catalase enzyme, and they are basically free radical scavenger. They are free radical scavenger cells, and inside they are the free radical scavengers H2O2, and inside it they are activating the catalase activity inside the peroxisome. Next one is reductase enzymes. They require NADPH for their action, and we can see these all 6 types of classes inside the first. Next we are going to see what are the first class or first subclass of the oxidoreductase. The first subclass of the oxidoreductase is dehydrogenase basically. And what does it mean, dehydrogenase is it? Dehydrogenase is basically the removal of hydrogen, the removal of any hydrogen from one substrate, the removal of hydrogen from one substrate and donate to the another. It is true. The removal of hydrogen from the some other substrate and giving this hydrogen to the another substrate, but what the another substrate is here is fixed. The another substrate is basically B complex vitamins And B complex vitamins like B1, B2, B3, B6, B5, and B9, B12, like this. These all are the B complex vitamins. So these all B complex vitamins, their coenzymes or the cofactors are present. Coenzymes are like this: NAD, FAD, like the NAD, nicotinamide, adenine, dinucleotide, like the FAD, flavin, like this. These all vitamins are gain some hydrogen donated by the some substrate, and this is called a dehydrogenase. Dehydrogenase, it means the removal of one hydrogen from the one substrate and giving to the another substrate, and here another substrate is fixed. That is vitamin B complex usually. And we can see in the first example here, the NAD plus is gaining one hydrogen and they are in the NADH state. So there is a gaining of hydrogen from they are taking from another substrate and giving to it. Next one, FAD is a source of vitamin B, and here vitamin 3 we get NAD, and this FAD is converted into FADH2. Here is the also gain of hydrogen, we can see clearly. And it is the, we can also see the NAD is the most common reaction here in our body. NAD is most common, most favorable reaction, because it's gain hydrogen easily, and FAD is converted into FADH2. There are also several examples where it's located in our general life and where it's again doing their function in our body. In the Krebs cycle or TCS cycle, tricarboxylic acid cycle, we can see the succinate dehydrogenase enzyme. And you know what we are discussing now, the first subclass of the oxidoreductase, and the name of it is oxidoreductase, that is the dehydrogenase. So succinate dehydrogenase and acyl-CoA dehydrogenase, these are the two enzymes where FAD is acting as a coenzymes, and they have a dehydrogenase activity. So they are the FAD and NAD. Next one is NADP. NADP plus is converting into NADPH. Here is the also gain of one hydrogen. They taking from the one another substrate to the going to vitamin B, and where we can see these reactions. NADP plus is converted into NADPH. Basically, they are seen in first two reactions of the HMP. So these two reactions of the HMP pathway, or the pentose phosphate pathway. HMP means pentose phosphate pathway, we can see. And what is pentose phosphate pathway, we discuss later everything. But for now you have to memorize these are uses in the first two steps of pentose phosphate pathway or HMP pathway. And next one, the cytoplasmic. In the cytoplasm, isocitrate dehydrogenase enzyme is present where this NADPH is acting, and the malic enzyme. So these are the three examples of it where it's located. And these all examples are important here in our general life, so you have to memorize it. The subclasses of oxidoreductase, first dehydrogenase, and you know now. Next one is oxygenase. There is a directly addition of oxygen to the substrate. Here we can see in the first portion, we are going to discuss this one by one. First we discuss dehydrogenase, next we are going to see about the oxygenase, and in the oxygenase, you know now, there is a directly addition of oxygen to the any substrate. When we are going to add, suppose that here is the some substrate here, and we are going to add oxygen. We are going to add oxygen molecules or the atoms here, hydrogen atoms here is going to add on it on the substrate directly. When the one atom is added, that one is called as monooxygenase. When both atoms of the oxygen is going to add, one atom and the both atoms of the oxygen is going to add, then they are called as dioxygenase. So you have to memorize, when the one atom is going to add on the substrate, it is monooxygenase, and the both atom is going to attach, then they are called as dioxygenase. So it will be clear for you. Now the one atom is added, or the both atoms of O2 will be added. These are called as monooxygenase and a dioxygenase. What happened to the monooxygenase is it? The monooxidase is having mixed function. What they are going to perform in our body, the monooxidase, what is the oxygenase is attached with one oxygen. What is the role in our general life bodies? We can see it is basically present in the most of the hydroxylase and cytochromes. What are the cytochromes are present in our body, or the hydroxylase in which these enzymes they are located, and their function is tyrosine hydroxylase enzyme, phenylalanine hydroxylase enzyme, tryptophan hydroxylase enzyme, 7-alpha hydroxylase, cytochromes. These all are the example of monooxygenase. Now we can see what are the examples of dioxygenase. Dioxygenase is the example is having tryptophan dioxygenase, that is also called as pyrrolase, and the homogentisate dioxygenase. So these are the two examples here, tryptophan dioxygenase or the homogentisate dioxygenase. And here we can see 5 examples, so you have to memorize it, in which monooxygenase and the dioxygenase and combinedly called as oxygenase, what are the reactions are coming in it. Directly questions are coming, oxygenase reactions, which of the flowing as a monooxygenase, so you have to memorize it. Next one is oxidase, that is second group. We can see here oxidase, there is a oxide something, and it means the removal of hydrogen from one substrate and donate to the oxygen. Here is fixed. In first what we discussed, we can see here in the dehydrogenase subclasses of the first group, we already see there is a removal of hydrogen from one substrate and giving to the B complex vitamin. But in the third we can see here oxidase, there is a removal of hydrogen from the one substrate and giving to the especially only oxygen. And when we see in the electron transport chain etc, there is a same activities happening. And what is it? Cytochrome C oxidase is taking the hydrogen from one part and supply to the O2. Next, in the complex 4 we discuss everything in the bioenergetics section. For now you have to memorize it. Or also in the monoamine oxidase breakdown of neurotransmitter it's used. So it will be also happen when the monoxide is working. Monoamine oxide, it is basically breakdown of norepinephrine neurotransmitter is done by it, or also enzyme is there, comet. We can see now second group of our enzyme. First we discussed, that is oxidoreductase OTH and T, that is translocase. In second group is OT, that is transferase. And what happened to this transferase? There is a transfer of something. So what is the transfer? They transfer the functional group from one substrate to another substrate. Suppose that here we see one substrate and here is the another substrate, and the functional group is attached here, and this functional group is transferred to the another substrate. So what is transferred here? Functional group from the first substrate to the second substrate. What are these type of enzymes is it? What are the subclasses of these enzymes here inside the transferase? The first we see trans is the any enzymes you can see there trans, it means these all are the included inside the class transferases. And what we discuss first, trans and trans, it means these all are included into the transferase family. And the first one is transaminase, which functional group is gets transferred here, amino group. Next aldolase, then aldol group, keto, and then methyl group. These all functional group will be gets transferred somewhere in the another substrate. And in the any enzymes with trans in its name, it means these all are belongs to transferase class of the class of enzymes. Next we can see inside the transferase another classification, that is kinases. Kinases is also included inside the transferase group. And what is transferred here? A special type of group, that is the phosphate group. There is a transfer of phosphate from organic phosphate molecule. The transfer of phosphate from the organic phosphate molecule means basically ATP. ATP is organic form of phosphate, and we can see the several example here how the kinase enzymes are working. We can see the glucose is converted into glucose 6 phosphate. Glucose is basically 6 carbon compound or the hexose sugar. What happened to it when they are going to gaining of phosphate on its 6th position? It means there is something added on it. It means something transfer in their structure. And what is transfer here? The phosphate group. And where from this phosphate group is coming? From the organic substrate. Already written there कि this phosphate group are coming from the organic, some organic substrate, and the organic substrate is here ATP. We can see here there is the 1 ATP is converted into 1 ADP. The triphosphate is converted into diphosphate. It means the one phosphate group is going to be transferred here, and the transferred phosphate we can see here. The transferred phosphate is here, we can see, and the magnesium is required for catalyzing these reactions. So what is reaction is occurred here? This reaction is catalyzed by the magnesium and also glucokinase enzyme. We can see that the glucokinase. Now from now when we see any kinase enzyme, then directly in your mind there is a organic phosphate somehow is going to added somewhere on the substrate. So next point we can see the glucokinase. Kinase means the addition of phosphate. And next point, magnesium is used for the catalysis reaction. Next we can see the types of hexokinase. The types of what are the different types of hexokinase are working here. It going to add the phosphate group on the 6th position of the all the hexoses. What the hexoses, you know, hexose sugars basically, glucose, fructose, these all are the hexose sugar, and this hexose sugar on its 6th position fixed. These all the phosphate group are going to attach on the 6th position of it, and so these all are called as glucose 6 phosphate. Hope it will clear for you. Next example we can see the fructose 6 phosphate is converted into fructose 1,6-bis phosphate. Here what is happening? The already one phosphate is present at the 6th position. So here we are going to attach the phosphate group on the another part, and that is on the first part of the fructose 6 phosphate. That is 1 ATP is converted into 1 ATP, means one organic phosphate will be going to attach here, and the enzyme is phosphofructokinase. Kinase, once again, it means somewhere the phosphate group is going to be transferred. Kinase means basically the transfer of phosphate group from one substrate to the another substrate, and the substrate we can see here. Basically in the hexose sugars they are going to attach on the 6th position. Next we can see phosphorylase. There is a third subclass inside the second class, that is the phosphorylase. There is a transfer of inorganic phosphate, not organic. Now we are going to see inorganic phosphate, and we can see this inorganic phosphate is going to attach when there is a glycogen. Glycogen is going to break. The breakdown of glycogen is done by the glycogen phosphorylase enzyme. Synthesis is done by the glycogen synthetase. The glycogen phosphorylase is going to break down of this glycogen, and they produces basically glucose. And what type of glucose? Glucose 1-phosphate is produces. They produces, we discuss everything in the glycogen metabolism. For now we have to memorize it. The glycogen is converted into glucose 1-phosphate when and there is a glycogen metabolism is happening. Then breakdown, which enzyme is acting there? There is glycogen phosphorylase. And what a phosphate coming from where? Because the glycogen converted into 1-phosphate. It means the phosphate comes from the somewhere, and where they comes from, that is inorganic source. And the inorganic source, that's why they are called as inorganic phosphate. So this is phosphorylase basically, and the glycogen phosphorylase action we can see here. Next we are going to see class 3rd enzyme, and the class 3rd enzyme OTH, that is the hydrolase. Hydrolase is basically breaking down of covalent bonds, and covalent bond is a strong bond. These covalent bonds is going to break down. And how the covalent, what are the different types of covalent bond? Do you know? The carbon, carbon, nitrogen, and carbon, oxygen. These are the CO, CC, and CN. These all the bonds are covalent bonds, and how we are going to study here, that is hydrolases. When the water is added, these bonds are going to break. It means what happened? When these bonds are formed somewhere, anywhere in the compound, we are going to add water there, and these bonds are going to break. So we can see here these covalent bonds are going to break by adding of water. It is true. And what happened? Where these covalent bonds are present, these covalent bonds are present in primary structure of all the macromolecules. And what are the macromolecules in our daily life? What we know about the macromolecules we discuss here, and how these macromolecules are containing covalent bonds, and how we are going to add water and these covalent bonds are going to break. We are going to see here in a table form. The first we can see the macromolecules. What are the macromolecules? We know that is the carbohydrate source, and the carbohydrate, that is protein. These all are the macromolecules, basically the nucleic acid and the lipids. Our food sources will be these all are the macromolecules: carbohydrate, proteins, nucleic acid and the lipids. In the carbohydrates there is some covalent bond. And what are the different types of covalent bonds we are going to see in the carbohydrate, proteins, lipids or the nucleic acid? In the first section we are going to see here, there is a carbohydrate, what type of covalent bond? That is glycosidic bond. And we have to know by heart, glycosidic bond is present into the carbohydrate structure, and they are going to break by the hydrolase enzyme, and it is a type of covalent bond. Next, in the protein, peptide bond is a type of covalent bond. In the nucleic acid, phosphodiester bond is a type of covalent bond. In the lipids, there is ester bond. So you have to know by heart these bonds, and these bonds are covalent bond. These bonds are present in the macromolecules. And what are the hydrolase enzyme? It's going to break these bonds. So we can see these all are found in our digestive system, basically. And in our digestive system, you know how the digestive system is going to work there. There are different types of enzyme releases, and these all enzymes are going to acting on these food particles what we eat, that is carbohydrate, protein, nucleic acid or the lipids. They are the food sources, basically, and these enzymes are going to action on it, and they are going to perform the action on it. So we know it, these hydrolases are going to break our food particles, basically the macromolecules what is present on the stomach or the intestine. So we can see in the carbohydrate there is several types of enzyme is acting, and the name of these enzyme are amylase, maltase, lactase, sucrase, lactase, maltase, amylase. These all are the enzymes, and basically many more. They all are going to acting on the carbohydrate source. Next one, on the protein source, there is one enzyme is peptidase, protease, and proteases are the several types like the trypsin, chymotrypsin, elastases. So these all are the enzymes. They are going to work on the protein, and they are going to break the what bond? Peptide bond. Next one is nucleic acid. What type of bond is covalent bond? That is phosphodiester, and they are going to break down by a special type of enzyme, that is nucleases. These nucleases are going to break down which bond? Phosphodiester bond. In where? In the nucleic acid. And what are the nucleases? Do you know? Endonucleases and exonucleases. What does it mean? The endonucleases are going to break down the nucleic acid inside it. It means in the inner part, and the exonucleases are going to break at the terminal ends. That's why their exo means external part. They are going to cut or the breakdown. There is phosphodiester bond. Next, the lipids. There is ester bonds are present, and these ester bonds are going to break down through the esterase enzyme or the lipases. So this is all about our macromolecules, and they are breaking the class 3 hydrolases. Different types of covalent bond, you know how to break it. And now next one is we are going to see other hydrolase classes. What are the other hydrolase enzymes? We have to know that is arginase. We discuss in the urea cycle, and in the urea cycle there is arginase enzyme is used, and what is the role of it? We discuss everything there. Next one is phosphatase. What is the role of phosphatase enzyme, and in which group the phosphatase are comes? We have to memorize it. Phosphatase are come under the hydrolases. Now the glucose 6-phosphate, we can see one example here, is converted into glucose. What happened? It means there is a removal of phosphate. We can see here basically phosphatase is the glucose 6-phosphatase enzyme. In the presence of glucose 6-phosphatase enzyme, the release of one phosphate here, addition of water and release of phosphate, and this release of phosphate is basically phosphatase. And arginase in using urea cycle. This all we discuss everything in the different parts of our biochemistry. So it is the basic, and we use these all reactions in the different parts of biochemistry, so must to memorize by heart. Next, fourth group OTH, that is and L. L is the liases. Now we are going to see liases. They are breaking covalent bond. Once again, they are breaking also covalent bond. How? Without adding water. Previous one, they are going to break covalent bond by adding the water, and that is hydrolases. But here is liage. They are not going to break covalent bond by adding water. Then what is happening here? Let's see some example. Fructose 1,6-bisphosphate is converted into glyceraldehyde 3-phosphate and DHAP. DHAP is basically dihydroxyacetone phosphate, and there is forming two components. We can see here, one is here and one is here, that they are forming two components. And what are it? They are the glyceraldehyde 3-phosphate and DHAP. These are going to break down. How they are going to break down? The fructose 1,6-bisphosphate is a 6-carbon compound, and they are breaking down into two parts. That is, the one is 3 compound and one another 3 compound. They are basically forming the two compounds. How they are going to forming two compounds? The 6-carbon compound breaking into two parts. What type of enzyme is it? That is liases. They are going to also break the covalent bond but without adding water. And what is the enzyme is here, which is going to break down by the lases, and these are basically our aldolase. The aldolase is the enzyme which is responsible for the breaking down of 6 carbon into 3 carbon fructose 1,6-bisphosphate into the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So in this section, what we are going to see in the 4th class, liases, they are breaking the covalent bond by the without water adding. And what is it? That is aldolase enzyme is going to break down here in the first case. And also several examples we can see. The any other enzymes, their enzyme name is having end with, or the name is having liases. We can see if the any enzyme with liase ending. So these all the enzymes are included inside the liases group. We can see in the liases group. And what are the different types of enzymes? We have to memorize it. That is HMG-CoA liase, HMG-CoA liase, that is coenzyme A, and the arginosuccinate liase and the ATP citrate liase. These are the 3 enzymes. They are ending with liases, we can see clearly, and these all ending of liases end with the names of liases are going to perform their action. And how they are going to perform their action? Breaking covalent bond without adding water. Next point in it, there is a enolases is also the example of it. And how they break and make, it means they are doing both the function. They are either breaking or either making the double bond. They are going to break or make the double bond by the atom elimination. They are going to eliminate something, and that is the atom here. And in the enolases, we can see they are going to eliminate something. So we will see some examples here. 2-phosphoglycerate, it gets converted into phosphoenol pyruvate plus H2O. So what is eliminated? Atom here, that is the H2O, we can see. And what is going to initiate this example, catalysis with this example, that is magnesium dependent. This example is we can see that is 2-phosphoglycerate in the phosphoenol pyruvate plus H2O is basically elimination of H2O atom here, and we can see they are the magnesium dependent reaction. And what happened in this magnesium reaction? What is the enzyme is it here? That is the enolase. Enolase is the enzyme here which is converting it, and the magnesium is required for the catalyzation of this reaction. So there is a making or breaking. We can also see these reactions may happen in the opposite direction. So they are the making or breaking of this covalent bond. So you have to must know by heart the enolases, liases, and the first one aldolases. These all are included into the liases group. So directly questions are coming. Aldolases are what type of, in which class of our enzymes they are included? They are basically liases. Next section we can see that is fumarases. Fumarate is also type of liases. They converting the fumarate into L-malate. They are going to convert. Fumarate is an enzyme. It is also included into the liases group in our 4th class. and they converting the hydration fumarate into the L-malate by the enzyme fumarase and by the addition of H2. And hydration reaction is set up the another oxidation. In this reaction there is another oxidation is occur. So leave it for now. We discuss everything in the different pathway, different metabolic pathway of the biochemistry. But we have to know by heart the fumarase reactions, what they are going to convert, and what they use their catalysis reactions, what type of enzymes, in which class of enzyme they are belonging to. Next one we can also see here, that is aconitase and decarboxylase. These are also inside the liases. We can see they are also included the liases in the class 4 of our classification of enzyme, that is aconitases and decarboxylase. The decarboxylation is happen basically two types: simple decarboxylation and oxidative decarboxylation. And this is the most important part of our enzyme here, the decarboxylation part and synthetase part. So decarboxylation is basically included inside the fourth class of our enzymes, and in which what different types of simple decarboxylation do you know? There is a question directly coming. What is the simple decarboxylation? It means the simple decarboxylation, it means the basically there is a conversion, what the histidine is converting into histamine. Simple. Histamine is a neurotransmitter, as you know. Glutamate is amino acid, they are converted into gamma aminobutyric acid. They are also neurotransmitter. Now the tryptophan is converted into tryptamine. So these all are the after done decarboxylation or the lysis activities, they are converted into something, and this is done by after simple decarboxylation. And what is the main coenzymes and what they are liberating after the simple decarboxylation is it? In all these reactions, we can see here, in all these reactions they are liberating CO2. One CO2 will be liberated, and there is what is the coenzyme is used here, that is PLP, pyridoxal pyrophosphate. We can see pyridoxal phosphate here, pyridoxal phosphate. Pyridoxal phosphate is the coenzyme we are going to use here for the conversion of all these simple decarboxylation in all the reactions. It not happen only in the tryptophan to tryptamine. In all reactions there is a removal of CO2 and coenzyme PLP will be used for the simple color. Now next we can see oxidative decarboxylation. In oxidative decarboxylation there is several examples. Pyruvate dehydrogenase, these all examples are very important. There is alpha ketoglutarate dehydrogenase, branched chain keto acid dehydrogenase. So these all are the three examples of oxidative decarboxylation. So what happen in these reactions? There is a use of a special type of coenzyme, that is NAD, and you know NAD is converting into NADH. It means there is a transferase of what? H plus from one substrate to another substrate, and the substrate is here dehydrogenase. We see already in the above, in the class 1 oxidoreductases, they are doing like the same activity, and that is also called as oxidative decarboxylation. There is also we are going to do the NAD is converted into NADH in the dehydrogenases. In the first class there is a first subclass of the first class. We discuss oxidoreductases, and that is doing the same. Next we are going to see the class 5th, that is isomerase, and they are initiated by the catalyzed by the isomerization reaction. So these are catalyzed by the isomerization reaction. And what are the different types of reactions is included inside the isomerases or the isomerase enzymes is going to act here? In the first we can see the glucose 6 phosphate, glyceraldehyde 3 phosphate, D-glucose, glucose 6 phosphate. So these all are important. Glucose 6 phosphate is going to convert into fructose 6 phosphate in a special type of enzyme, that is isomerase. Okay, why? These two, glucose or fructose, these are the isomer to each other, or these are the isomerases. What does it mean? They are the isomer to each other. So we are going to change, or the different types of enzymes that is isomerases, are used to their conversions. That's why they are called as the isomerases. You can also see glyceraldehyde 3-phosphate is converted into DHAP, dihydroxyacetone phosphate. These are also isomerases. D-glucose is converted into L-glucose, and these are racemase. These are the racemic mixture to each other. And the glucose 6-phosphate is the converted into glucose 1-phosphate. They are mutases. They are the phosphoglucomutate. So we have to memorize what are the enzymes used in the conversion of different types of reactions, and these reactions we use in the different metabolic pathway. I already told you. So you have to learn by heart, because when you know these all enzymes and what they are going to convert and in which class they are belonging to, so it's very easy for you to learn biochemistry, because in biochemistry we are going to see these reactions in a different cycle. So that time you can imagine and think which enzyme is belonging to which classes of group, and what is the subtype, and what is the main role of these enzymes, what they are going to liberate, what is the coenzyme of it. So it's very easy. And these catalyzes reactions, in these all the reactions intramolecular transfer of phosphate is occur. Internally there is somehow the transfer of phosphate. In all these isomerases reactions, or we can see basically in these reactions, glucose 6-phosphate is, you can see in the same glucose, the phosphate position will be changed. First, initially the phosphate will be present at the 6th position, later on they are present on the 1st position. It means in the same molecule, that is intra, and intramolecule, same molecule, there is exchange of phosphate from the 6th to 1 position. That is done by the mutases enzyme, and these are called as phosphate group is transferred where in the molecule glucose, and the transfer what phosphate, that is called as same mutase, that is phosphoglucomutase. So it's going to transfer the phosphate in the same molecule. Next we are going to see the class 6. The class 6 name is ligases. Ligases is class 6 here, and ligases is going to join or make the covalent bond. Either they make the covalent bond, they are going to making the covalent bond, and we also break the covalent bond in the previous part, hydrolase enzyme, and the joining two substrate. Ligases are going to bind something. Suppose that this one is one substrate and this one is one substrate. These both the substrates are going to attach. These both the substrates are going to join together. So they are joining or the attaching. These ligases are done by the class 6, that is ligase group, and they also making the covalent bond, and there is a ATP is required. It means there is energy requiring process. All the ligases are energy requiring process, and we can see what are the different types of subclasses or the enzymes or the reaction which happen inside it. The first one is carboxylase enzyme is acting there, and the synthetase, something is going to be carboxylase means carboxylation, and the synthesis is occur here. In all the carboxylase enzymes, what they are working here, in all the reaction they are adding, requiring, or the coenzymes are using, what they are doing in all the reactions, there is a CO2 is added. It means one carbon is added, and ATP required. ATP is given to these reactions, and coenzymes in all these reactions will be the biotin, and catalyzed by the ligases enzymes. So in all the carboxylase, what we discuss now in the examples, in all these carboxylase reactions, there is a addition of one CO2, and we can also see there is a ATP is required, biotin is a coenzyme, and all the ligase enzymes will be used here. In the first few examples we can see these all are important here. Basically pyruvate is converted into oxaloacetate. In the first we can see here the pyruvate is 3 carbon compound, but they are converted into 4 carbon compound. What does it mean? There is addition of 1 carbon is must, that's why they are converting into 4 carbon. And here we can see the CO2 is added. It means CO2. It means 1 carbon is there and they are going to add. And what is the enzyme? Is it carboxylase? Wow. Because we already know that in the carboxylase 1 CO2 is added, biotin is a coenzyme, these all are ligases enzyme is present there. So we can see everything, and these biotin molecules are present. And what is the name of the substrate is pyruvate, the pyruvate carboxylase. It's simple. So we add pyruvate in the initial structures, and that is the enzyme here. In the next section you can also see propionyl CoA, acetyl CoA. Acetyl CoA is converted into the malonyl CoA. The two compound converted into three compound. Once again there is an addition of one carbon, and the enzyme is acetyl CoA carboxylase. Carboxylase, why? There is a carboxylation, addition of one carbon. Propionyl CoA carboxylase, addition of one carbon, ATP, and in all the reactions biotin are used. Next we can see what is the independent role of it. The biotin independent carboxylase reactions. In some reactions the biotin is not required for the carboxylation reactions. What is it? We have to learn by heart these all reactions what we discuss in this section. The carboxylase synthetase, these all reactions are very important in the biochemistry. So you have to know where these enzymes, what are the coenzymes, what are the ATPs requiring or not. So these all are important. In the next biotin independent carboxylation. In the carboxylation reactions there is no biotin is required. So what are the reactions is it? CpH1, CpH2. What does it mean? CpS, carbamyl. We can see carbamoyl phosphate synthetase. It means it is a enzyme, that is carbamyl phosphate synthetase 1 and carbamyl phosphatase synthetase 2. These are the two enzymes which is going to perform their action without they are going to use the biotin. Also in the gamma carboxylation there is no use of biotin, and the addition of sixth carbon in the purine ring, that is basically called as AIR carboxylase, and the malic enzymes. In all these steps we are going to not use the biotin. That's why they are the biotin independent carboxylation state. And in the previous pyruvate, acetyl CoA, or the propionyl CoA, in all these enzymes we are going to use, in all these reactions we are going to use the biotin, ATP, CO2 added, and the lyase enzymes. Next section is synthetase. First we already discussed about carboxylase. Next one is synthetase. Their ATP is required here, yeah, but what we discussed in the previous section, there is a biotin, CO2 is added, but nothing to going to add here. There is a glutamine synthetase is one example, and one is the carbamyl phosphate synthetase. These both reactions are important, or we discuss later. These both enzymes we are going to use in the different cycles, but for now you have to memorize these are the synthetases. They are belongs to class 6, and the synthetases do not synthesis. Here we discuss synthetase. Synthetase means they require ATP, but synthetase, somehow the group synthase belongs to class belongs to all the classes. They belong all the classes except 6. What I'm going to tell you, here the carboxylase and synthetase are included into the class 6. It's good, but synthase are belongs not to the 6 classes but all the other classes. So it means they belong to the 1st, 2nd, 3rd, 4th, 5th, but they're not in the 6 classes. Synthase, and they do not require any energy, so there's no use of ATP. And what is the main role? Now you tell me, what is the synthetase examples? That is glutamine synthetase and carbamyl phosphate synthetase. Now we can see here the type 7. Basically the type 7 is translocase. I already told you, translocase means the transfer of molecules or the ions across the membrane. We are going to now transferring the either molecules or either ions across the membrane. Some membranes they are crossing. They are called as ATPase activity. Earlier these all activity, translocase activities, are called as basically ATPase, and they belongs to class 3rd previously. That all the translocase enzymes are previously belonged to 3rd classes, but for now they are classified into separate classes. So these all are the enzymes, general basic knowledge how the enzymes are going to perform, what are the different reactions in our metabolic pathway we are going to discuss in the later stages of the biochemistry. And we know by heart the carboxylase, synthesis, or the first class, second class, third class, what are the different classes of the biochemistry or classification, what are the reactions, what are the coenzymes required, are the ATP requiring or not. So these all steps we have to learn it, and it's very easy to memorize it, because when we memorize one time, we are going to applicate in the different section of biochemistry. So you have to memorize in one time at a once, and we have to learn by everything in the different section. So thank you. We will meet you in the next section in gymology of the biochemistry. Thank you. See you. Bye bye.